Thank you very much to the organisers for inviting me here to Dubai, and also thank you for providing such wonderfully comfortable couches in the front here. It's been a pleasure to sit there. Okay, so over the next 25-30 minutes I'm going to tell you about what I think is the next important question in molecular and cellular biology and human genetics, and that is linking DNA variation to changes in gene expression. And the story really starts here in 2001 when the first draft of the human genome was published, and it told us that there were 20,000 genes, and we've subsequently learned that that genome is highly variable between individuals of the human population. And perhaps the greatest molecular biologist of our age, Sidney Brenner, said that getting the sequence was the easy part, as only technical issues are involved, and the hard part is really trying to crack that code and find out what the sequence really means and how it's executed. So in my own lab, what we try and do is work out how mammalian genes are switched on and off during lineage commitment and differentiation, how those genes are perturbed in human disease, both inherited and acquired disease, and the ultimate goal of this is really to try and manipulate those switches to improve human health. So our philosophy is that we work on a single gene locus, and we try and understand the principles by which genes are switched on and off, and then we apply that to genome-wide data, all the 20,000 genes, to see if the principles that we're learning apply equally to all of those other genes, and thereby we can crack how these multiplicity of genes are switched on and off. So we have to work on a very defined cell system, and the system we work on is erythropoiesis, which starts off with a hemopoietic stem cell, which then undergoes commitment to the erythroid lineage, and then goes through differentiation right the way through to forming mature red blood cells. Now, we also work on the gene that's most prominently expressed during this process, the globin gene, which is switched off in a hemopoietic stem cell here, but it gradually gets switched on during erythropoiesis and then gets switched off again right at the end of erythropoiesis. So it's a very interesting and typical differentiation switch. And the beauty of this system is that we can isolate hemopoietic cells at various stages of this process using high-speed cell sorting, and that enables us then to get snapshots of what's happening with gene expression as a hemopoietic stem cell goes through the commitment and differentiation programs. Now, we study this both in the human and mouse, and so for the point of view of this talk, I'm not going to differentiate between them because we now know that the human and mouse clusters are both regulated in very similar ways. Now, this just reminds you of the simple genetics of hemoglobin. There are two gene clusters involved, the alpha genes and the beta genes, which produce alpha and beta globin to produce the heterotetramer, hemoglobin A. And we study really just the alpha globin genes. All the work in my lab is to do with alpha globin expression. So here are the alpha globin genes. This is the mouse cluster, so they're on chromosome 11. And although the genes are expressed in a highly tissue and developmental stage-specific manner, what you can see is that they're actually surrounded by widely expressed housekeeping genes on either side. And the question we've been interested in is how much of the chromosome do you actually need to get fully regulated expression? And we've found that this region here, outlined in pink, is what you need to get fully regulated expression. Doesn't matter where we put that back in the genome, it's regulated properly. And so this was an experiment that we did a few years ago where we used chromosome engineering to replace that segment and put it anywhere we like in the genome, and it works perfectly well. And that segment's sufficient to produce fully regulated gene expression. So the next question I'd like to try and address is, if that's the unit that you need to get fully regulated expression, where are the key regulatory elements within that that drive globin gene expression. So I need to diverge a little bit just to explain something that was discussed earlier on, and that is that you all know that to get 2 meters of DNA packaged into the nucleus of every cell, it has to be tightly packaged with chromatin. It has to be wrapped around nucleosomes so that you can pack it tightly into the nucleus. And what this shows is the chromatin here packaged with the linker histone H1, which is the green circles. And the histones, as we've heard again, are modified in different ways, and these histones in this highly packaged chromatin are actually methylated. So now, to get a gene expressed, it has to be unpackaged. You take away the H1 histone linker, and then it unfolds to produce this structure that you saw before, which is called the 10 nanometer fiber. And these little lollipops I put there to indicate the N-terminal tail modifications. So now, if you treat those, what you need also to get gene expression is another step, and this involves the chromatin remodeling factors that you heard about earlier, and they are required to use the energy of ATP hydrolysis to either remodel or remove nucleosomes and then allow transcription factors to get access to the DNA that they're going to activate. Now, this is all you have to know, that in actual fact these elements are the regulatory elements in the genome. So we can actually identify them by the fact that they are particularly sensitive to nucleases, and they'll cut where the transcription factors are binding the DNA and they've moved the nucleosome out of the way. And it produces, on a southern blot we can see that by this subband that we can see when we digest with DNase 1. And these are called DNase 1 hypersensitive sites. Well, nowadays we can identify all the DNase hypersensitive sites in the genome by using DNA sequence analysis, where we lightly digest nuclei with DNase 1, and then we can just sequence the products that correspond to the DNase 1 hypersensitive sites. And this is what it looks like. So this is part of a genome-wide study, and now we're just looking again back on the alpha globin cluster. And what you can see are the DNase 1 hypersensitive sites, which are the peaks on these diagrams, and they correspond to where the regulatory elements are that are controlling globin gene expression. And we can divide them up between the ones that are associated with promoters that are modified by this particular chromatin modification called H3K4ME3. It doesn't matter about the detail, but they point out where the promoters are. And then there we are. If we want to find the enhancers, they're modified by another modification called H3K4ME1, and that enables us to identify the enhancer regions. So now we've got the promoters and we've got the enhancers, and so we can do that throughout the genome. And we also find that the enhancers are where you're binding erythroid-specific transcription factors such as SCL. Okay, so now the enhancers are what determines when genes come on and which cells they come on in. So if we look at here, this is the enhancers in an erythroid cell marked by the red arrows, and the 2 promoters of the alpha genes marked by these 2 red arrows, here, here, and here. And what you can see is that in a non-erythroid cell, when the globin genes are not activated, there are no hypersensitive sites. So what it's saying that in an erythroid cell, those particular regulatory elements are now binding transcription factors and becoming activated. So what we can also do is now we take cells that are going through from a hemopoietic stem cell through to a red blood cell, and you can see that early on the hypersensitive sites aren't there, and then just before globin synthesis starts in this type of cell, the hypersensitive sites are formed and the complexes are formed. So this is based on a lot of data that we've produced over the last 10 years. What I'm going to do now is just summarize that data to give you a model of how we think a gene is switched on within the context of chromatin. So if we look along the top here, you'll see the cell changing. So if we look at a stem cell, there are no transcription factors bound, either at the enhancer or the promoter, and there are no chromatin modifications present at that stage. The first thing that happens is a pioneer factor comes in and it binds to the enhancer. And then if we go a little bit further, other factors come in to reinforce that binding. We go a little bit further now and other transcription factors come and join the party. And we get to this stage, and the next thing that happens is all the chromatin modifications, as a result of that transcription factor binding, are now in place. And if you look along the top line, what you can see is that that is occurring before globin synthesis has been switched on. So the next thing that happens is polymerase II, which is the enzyme that you need to make RNA, now binds at the enhancer, and using a technique that I'll show you a little bit later on, it's called chromosome conformation capture. What we now see is that the enhancer loops out, joins the promoter, and that is the cue to start transcription from the promoter. And what we've also found recently is that that also is the cue for the enhancer itself to be transcribed. Now, what we're going on to do in the lab at the moment is to try to make models that will enable us to visualize both the chromatin movements and the transcription and observe this in real time. So that's the next thing we're going to try and do to complete this model. So now let's move on to the sort of other aspect of what my lab does, and that is we look at natural mutations, the sort of things that we've heard about today, to try and inform our basic research. And of course, the interest of our lab is thalassemia, and as Errol pointed out, this is the world's most common form of inherited anemia. 300,000 people are severely affected every year, and of course, you heard that here in Dubai, that in actual fact, this is an extremely common form of disease. So what happens in alpha thalassemia when the alpha genes now are downregulated? Well, what happens is that you don't now form enough alpha globin to combine with beta globin, and therefore you develop an anemia, and as a result of that, you form excess beta globin chains, which form this new type of hemoglobin called hemoglobin H. Now, if you take some blood from a patient with alpha thalassemia, what you can see is that here, on this bit of the electrophoresis, you can see the hemoglobin H as a new type of hemoglobin. And what the hemoglobin does, because it's unstable, it precipitates in the cells and precipitates onto the red cell membranes, and it produces these abnormal types of red cells which are fragile and give rise to hemolysis and cause the phenotype we associate with thalassemia. So over the years, we've studied about 10,000 patients worldwide with this disease, and just to run through quickly, what we see is that there have been over 60 point mutations that have been identified. We often find that there are deletions involving one or both of the alpha globin genes, and then sometimes those deletions go right the way up to the telomere. They remove some of these other genes, but in actual fact, the patients, despite the fact they've only got one copy of all those other genes, remain completely normal, apart from their alpha thalassemia. But if the deletions go in the other direction, by the time you take out about a megabase of DNA and perhaps up to 100 genes, then you start to produce developmental abnormalities. And if you go far enough, then you bang into the genes that are important for tuberosclerosis and adult polycystic kidney disease, and it was mutations here that cause alpha thalassemia plus adult polycystic kidney disease that allowed our consortium to identify these important disease genes. Okay, so another type of interesting mutation that occurs are deletions that remove the regulatory elements but leave the alpha globin genes themselves intact. So if you just looked at the alpha globin genes, you'd think, well, everything's normal. But in actual fact, what's happened is you've made a deletion which has removed the regulatory elements. And these were the first types of deletions which told us that there is long-range regulation of alpha globin expression. So it really identified those sorts of distal enhancers that now are so common for many genes in the genome. Now, this was another interesting case of hemoglobin H disease from Melanesia. And what we found here is that when we sequenced the alpha-globin genes, they were normal, and we looked for mutations, deletions, whatever, and we found absolutely nothing. So we resequenced the entire cluster, and here we found that there was just one polymorphism which associated with this disease. And I'm going to very briefly go over this, and what that polymorphism, that single point polymorphism did was to create a new GATA1 binding site. So a new transcription factor binding site was brought in, and that's the cause of this disease, because what happens is that that recruits all of the other factors that I've told you about to create what we think is a new DNase hypersensitive site, which corresponds to a new promoter which is lying upstream of the alpha-globin genes. And so what we think happens is that that new promoter steals the activity from the upstream regulatory elements, and there's not enough activity left to drive alpha-globin expression normally. And we think that that might that's a gain of function mechanism by which regulatory SNPs may cause many human diseases, and it will be very interesting to see how many mechanisms fall into this category in the future. So variants of the alpha cluster can affect long-range regulation of expression in the surrounding chromosomal landscape. This is the next point I want to make, and it really relates to this observation. So here are the regulatory elements up here lying in this gene, and they control alpha-globin expression, which is located 60 kilobases away down the chromosome here. Okay, so what we also found was that those regulatory elements control expression of another gene here called NME4, which is 300 kilobases away on the chromosome. Okay, so then we wondered, well, what happens to patients who have lost their alpha-globin genes? What happens to expression of this other gene that's located 300 kilobases away? And so we looked at that very carefully, and what we were able to show was that in these patients that had lost their alpha-globin genes, alpha-globin expression went down. That's what you'd expect. But what we also found was that expression of this gene down here went up tenfold. And so what we think happens is that if you lose the competition of the alpha-globin genes here, the activity of that regulatory element goes 300 kilobases along the chromosome and upregulates the linked gene on the chromosome 300 kilobases away. So that just tells us then that the variation enhancers and promoters may have very long-range effects on gene expression, and also the targets that are driven by those enhancers are not always obvious. So the next and the last part of my talk is really to say, do the principles that we've discovered at the globin locus inform us about the mechanisms underlying other forms of complex disease? In other words, we've looked at monogenic disease. Is this also what's happening in polygenic disease? So what's emerged over the last year or so is that whereas monogenic disorders often result from mutations in the gene itself, very often complex diseases are due to mutations in these distal regulatory elements. And so the trick now is to try and find out how those polymorphisms affect the cis-regulatory elements and thereby have an effect on the promoters of the genes that those cis-regulatory elements are linked to. And so we did a study in which we, rather than using a human population, we wanted to use a good experimental model to answer this question. And what we did was to use 8 inbred strains of mice that had been fully sequenced, phenotyped, and wondered if they showed variation in hypersensitive sites. So this, again, shows you that we've done the same experiment now. We've looked for DNase hypersensitive sites, and we've looked for them in 8 inbred strains of mice. They all vary from each other in terms of their sequence and their regulatory elements, but within the inbred strain, they're identical on both chromosomes. And what you see is that if we do this experiment, it's very reproducible, and all of the 8 strains show no variation within the alpha-globin cluster. However, what we found was that about just 1,500 or so of the peaks were variable. So let's just look at that. So what we're seeing here is variability in the DNase hypersensitive site. So here's the DNase experiment now that's been done on 8 strains of mice. And what you can see is that here the hypersensitive site is present, and then here the hypersensitive site has gone. And so it goes on down through the 8 different strains, either present or it's absent. And what we found is the presence or absence of the DNA's hypersensitive site is related to the underlying DNA sequence. So next we asked, is the change actually associated with a change in gene expression? So we're seeing changes in an enhancer. Is it actually affecting expression of the gene that's linked to it? So here are two strains of mice that haven't got a hypersensitive site, and what you see is the fact that they're not making any expression from the linked gene. But here's two strains of mice that do have a hypersensitive site, and in both cases their gene is switched on. So here the variability in the upstream region is affecting whether or not the gene downstream is switched on and off. And again, this is linked to the underlying DNA sequence associated with the regulatory element. Now the strange thing was that it was only 7% of these variable sites where we could see change in expression of a closely linked gene. So we wondered, was it going to be like in the alpha cluster where we could be affecting expression of genes 300 kilobases away? And so we asked the question, these elements regulate more distant genes. So what we now know from the literature is that these types of regulatory elements, they might actually lie within the introns of the gene that they regulate. They can lie downstream, and sometimes they can lie far upstream. And if we look at the literature in general, such elements can be 10 kb away, but they can be 1,000 kilobases away. So this is a huge distance in the chromosome. And not only that, but they can actually lie within the introns of another gene that they don't regulate at all. So this is a complex issue, and what we needed to do was to identify all the cis-actin elements that interact with a single genomic feature. So for example, if we take a regulatory element, we want to ask, well, which gene in the genome is it actually controlling? And that's what we've been working on recently, and this is the work of Jim Hughes in my lab. And so we're using this technique called chromosome conformation capture. And the basic principle here is that if you look at two regions that are separated, an enhancer and a promoter, if you make some oligos like this, they're never going to make a PCR product. But what we think is happening is that they are interacting with each other through multi-protein complexes to produce this sort of structure here, this sort of loop structure that I've described. So what we can do is we can cross-link that structure. So now all the proteins and DNA are stuck together. We then digest the chromatin with these restriction enzymes indicated by the arrows, and then we religate these under dilute conditions to produce these sorts of structures here. Okay? So what we can then do, and I'm not going to go into the details of this, very happy to talk with people about it after, is that we enrich these junction fragments from a library and then sequence them through high throughput sequencing. And this is the sort of result we get. So you can see that we're capturing now from the alpha globin cluster, from the alpha globin genes, and we're asking which elements interact with the alpha globin genes. And so what we see, in fact, is all the regulatory elements that I've shown you before are interacting with the promoter. So that's a lovely test using all the data that we know about, and it confirms, using this new technique, that we can identify all the regulatory elements that interact with the alpha globin promoter. And what's nice is that it's highly reproducible. You can see two biological replicates here, and also it's tissue specific. So in ES cells, where this interaction isn't taking place, we don't see any interactions with the upstream elements, but in erythroid cells, we do. Okay, so that's a single experiment, but now we do it high throughput, and we can look at 400, 500, I don't know how many, what's the upper limit. We've done 400 so far in a single experiment. Now, these data have been validated using the alpha globin cluster, but can we use this DNA sequence analysis and capture to analyze GWAS hits in complex disease? That's the question. And so I'm just going to show you one example where that's worked. So here what we did was we identified a gene called PNPO, and what we wanted to do was identify the regulatory element that is linked to that gene. And so we captured from the promoter here of the PNPO gene, and we asked what is it interacting with, and what you can see, it's interacting with this regulatory element here, which is associated with the DNase 1 hypersensitive site. So that was very nice, but what we also showed was that the sequence underlying that regulatory element is also polymorphic. It's changing in its DNA sequence. And so we wondered, do the underlying polymorphisms in this gene alter transcription factor binding, which we know is essential to form these regulatory elements, and change expression of the gene that is linked to it? And so we did one last experiment here, and this was due to the fact that we got 200 million reads for DNase 1 per strain. And what that enabled us to do was to map the frequency of cutting within this regulatory element. And what you can see is that the frequency of cutting is very high where there's no protein and relatively low where there is protein. And so this produces what we call a DNase 1 footprint. And if we look at that in reality now, here's the DNase 1 footprint, and you can see these two holes in the middle of the footprint, and that's where the DNA binding proteins are actually binding. We then looked at the sequence underlying that, and we saw two haplotypes which differ, and they're due to polymorphisms that actually lie in the region that is bound by the transcription factors. And so we called these haplotype A and haplotype B, and then we looked at the associated gene expression from this. We had a transcriptome analysis on all of this, and what we find is when you get haplotype A, the link gene, which is much further down the chromosome, is not expressed, whereas when you have haplotype B, which allows transcription factors to bind, the gene linked to it is now expressed. And this is going back to these 8 inbred strains of mice. So what we think is happening is that the enhancer here, when you have one particular set of polymorphisms, you don't form the enhancer, and therefore you don't get expression. But when you have another set of polymorphisms underlying this protein binding, you do get an enhancer form, and that is able to loop with its promoter and express the gene. Okay, so the conclusions are then that analysis of the globin cluster has established for us many of the principles by which enhancers activate promoters. And this has been done in the ways that I've shown you, by basic biology and by human genetics. And what we're now seeing is that many GWAS hits associated with complex disease lie in these cis-regulatory elements, but the genes that they regulate are not clear, and that's really the stumbling block in this field at the moment. So using the techniques which we've validated at the globin loci, we've shown that we can use a high throughput platform which allows us to link cis-regulatory elements to the genes that they actually regulate. And we've illustrated in this last little bit how the technique can be applied to link SNPs in the gene whose expression they influence using these trick of using inbred strains of mice. And so the next part of this is then to move this platform over to human genetics and see if we can identify the genes that are being regulated by some of the strong GWAS hits that we know to be associated with common diseases. So I think with that, I just want to thank some of the people that did this: Doug Verneman, David Garrick, Mona Husseini, and Jonathan Flint. And also the new technique was developed by a very, very good colleague, Jim Hughes. So with that, I'll thank you and answer any questions.
Thank you very much for this comprehensive speech. So we're open to questions now about all the previous lectures and the session. So I'm sure there are lots of people who want to ask questions. Yes.
Thank you very much for the lecture. I would like to ask Dr. two questions. First one, what makes the enhancer and the promoter stick together?
Yeah, that's...
And loop with that. This is my first question.
And it's a terrific question because that is at the center of understanding. So sorry, the question was, what makes the enhancer and the promoter stick together? And it's a great question, and it's really at the center of what enhancer and promoter biology, gene regulation is trying to answer at the moment. So the simple idea is that there are proteins in these two regions that have an affinity for each other. So you form a multi-complex here, a multi-protein complex here, and that probably these regions are moving about rather randomly, but they occasionally bump into each other. And when they do, the affinity is such that these two multi-protein complexes become stable and form a further multi-protein complex. And there's some very nice experiments that were done by Gerd Blobel just recently, in which he was able to make two pieces of chromatin stick together by simply recruiting proteins that interact with each other. So in other words, you stick a bit of glue here, and you put them together and they stick together. And it's a very beautiful experiment that was published in Cell about 9 months or a year ago.
What you look at the other part of the DNA loop.
Yeah.
There's no scheme like processing, RNA processing. Is there any sequence in the two parts?
No. So obviously, so the question was, what happens to the intervening loop? And so, and the comparison was, is it like splicing? Well, it's not like splicing because the loop itself is not removed. So it's still there. So it's like, you can imagine a piece of string, the two bits come together, and the piece of string is still hanging down. That's the sort of, these are probably very naive views of how it happens, but that's where we are at the moment. Yeah.
But since this time, we didn't talk about that.
Well, I've only got half an hour. I mean, I'd be happy to, if you want, I'll be happy to tell you about that. Yeah, I mean, I haven't told you about that. That's another completely independent project. And just to fill people in, there is an unusual syndrome in which alpha thalassemia is associated with developmental abnormalities, in particular mental retardation. And what that's due to is mutations in one of these chromatin remodeling factors that use the energy of ATP hydrolysis. There are mutations in that, and we think that gives rise to abnormal transcription, although we don't still know the mechanism by which that happens. And it's a complex story, but there's been a lot of progress, and it's a very interesting protein which is of relevance both to transcriptional regulation, and more recently it's been identified as a tumor suppressor. So it's a really interesting protein. Thank you for asking, but I'm sorry there wasn't time to discuss it.
Okay. Okay. Yeah.
I'll speak loud. So just a general question. Andre Joshi from Seattle, EMC Seattle. So is the complex law more generally that there's no annotation across the omics, from genomics to transcriptomics to proteomics? And what's your opinion on what can be a better way to
Yeah.
understand?
Yes, I think there are two ways to do it. I mean, one can obviously do association between if transcription goes up or down a gene and it's associated with polymorphisms in the enhancer, you might infer that this going up and down had something to do with the polymorphisms in the enhancer. I think what's very helpful about this, if you can do it in a high throughput way, you can make a physical linkage between those two, and you're not just relying on the statistical association. You can actually mechanistically show that those two sequences are interacting. So, I think that there are ways to do this bioinformatically, but I think you'll always be left with the question of whether that's a direct or indirect consequence of the polymorphisms.
The follow-up question is, with next generation sequencing, is GWAS necessary?
Is GWAS necessary? Well, yeah, I mean, I think yes, I think it is actually, because I mean, I think that essentially the GWAS part of it is just simply, can you link DNA variants to particular phenotypic traits? And I think you'll still need that, even if you have the whole genome. You still need the statistics to link up sequence variation with gene expression and phenotypic consequences. So, yeah, I mean, now, if you ask me whether it's worthwhile, the whole GWAS study is worthwhile, I could tell you about that for half an hour, but perhaps we could do that a later time, because I think it's a very interesting area for us, and it's a very challenging technical problem. I mean, I think we heard from David Cooper's talk this morning. I mean, just how complex these diseases are going to be to solve genetically when one's looking at these various patterns by which you can alter penetrance. So I think you've got that as another layer of complexity. So there's plenty of stuff for young people to do coming on over the next 30, 40, 50 years.
Okay. Any other questions? All right.
Oh, yeah. 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 So that's a super question. And after David's talk this morning, I said, look, if you want to study variable penetrance, I mean, sickle cell disease is such a wonderful example. So in sickle cell disease, everybody has the same mutation on both alleles. And so the question is, why is it that some patients with that die in the first 2 years of life, and other patients might live to the age of 60, 70, and not be diagnosed until they have a blood count, okay? They've all got exactly the same mutation. So there must be quite a lot of interacting factors that are altering the penetrance of that disease. And it also illustrates the fact that you can have lethal mutations and be perfectly okay, which is another point that David made. So all the work to look at variable penetrance in sickle cell disease has been going on ever since Linus Pauling first identified this as the first human molecular disease back in the 1950s. And there's a huge amount of data linking up genetic variation to sickle cell disease. And as a result of all that work, we know of about 3 things that really are important. One is the level of hemoglobin F that you produce. The second is whether you co-inherit alpha thalassemia. And there are other variable factors which are associated with white blood cell count, so on and so forth. But even if you look at a patient with all that knowledge, and you say, well, is that patient going to be severe or mildly affected? You can't do it. What you can do is say, as a group, this set of patients will be mild, and as a group, that set of patients will be severe. But on an individual basis, you still can't do it. And I think that's a really important thing for us to keep in mind with all of these diseases, that actually, despite the fact that we'll have the genetic modifiers, clinically, it still might be very hard to predict outcome. And I think that's important, because what it might be saying is that, in actual fact, it's really the genetic background in general that's important. Those are big modifiers, hemoglobin F and so on and so forth, but it's probably much more than that, and there's a huge amount of genetic variation which might be modifying the disease. Now, the final point of your question is whether the promoters and enhancers vary, and the most important determinant of hemoglobin F that's linked in cis, not in trans, but linked in cis, is a polymorphism in the promoter of the gamma gene, okay? So you're absolutely right, there are some cis polymorphisms, but also there are many transacting polymorphisms, of which fetal hemoglobin expression, transacting factors affecting fetal hemoglobin expression, alpha thalassemia, so on and so forth, are really important.
Okay, well, thank you very much. With this, we will end this session.